Welcome to ISL Enabled Virtual Lab of CSIR.
Hi, welcome to our Structural Biology Lab of CSIR CCMB. Here we try to understand two important but overlooked aspects of biology, that is chiral proofreading and lipid metabolism. Our primary focus centers on understanding the macromolecules through structure and analysis, but we also use a vast array of techniques to get a comprehensive analysis of the subject matter. The shape and structure of protein determines its function. We use cryo-electron microscopy as well as X-ray crystallography to reveal the same. To have a holistic overview, we use different model systems like bacteria, archaea, yeast, plant, mouse, etc. to test our hypothesis backed by a deep structural understanding. Interestingly, all these efforts are led by Dr. Shankar Narayanan, who, a physicist by nature, got intrigued by chemistry problem involving chirality and its nuances in biology and evolution. We also try to appreciate these processes from evolutionary standpoint. Our lab is actively researching on co-evolution of lipids and proteins.
All life forms are made up of cells, and these cells are encompassed by membranes. And membranes are made up of a class of molecules called lipids, and fatty acids are a major constituent of lipids. We study a class of proteins called fatty acid AMP ligases, FALs in short, that activates these fatty acids and transfers to a class of multi-enzyme complexes called polyketase synthesis and non-ribosomal peptide synthetases. These complexes are involved in producing natural products such as antibiotics, surfactants, lipopeptides, etc., to name a few. We have identified several mechanistic determinants that helps FAL achieve its remarkable specificity and able to produce specific natural products. And we are also interested in understanding the evolution of this protein across life forms. And in eukaryotes, when we have identified that there is a distant homologue of this FALs called DIP2, where it has completely changed its avatar and it's acting on a completely different class of lipids called diacylglycerols. And we have also seen that this protein inside the eukaryotic cell acts on a specific location called membrane contact sites. So these locations are places where any two organelles come together, interact with each other and exchange metabolites for communication.
For the past 22 years, our group at CCMB is trying to understand unique proofreading mechanisms that are operational in biological system to maintain quality control while translating genetic code. Amino acids are chiral molecules. They come in two flavors, L amino acids, D amino acids. However, proteins are made up of only L amino acids. Our major work is understanding how these D amino acids are kept away during protein synthesis. At CSIR CCMB, our work on proofreading is essential in understanding the precision and fidelity during protein synthesis at molecular level with implication on various fields of biology and biotechnology.